Hello, everybody. My name is Spencer Reiling, and I am an undergraduate student working in Dr. Woo-Suk Chang's lab. I am currently working with my graduate mentor, Sarobi Das, in her research concerning the herbicide glufosinate ammonium and its impact on our novel bacterium, Bradyrhizobium japonicum. B. japonicum is a nitrogen-fixing soil bacterium that is a common symbiont of soybean plants and has been used to reintroduce nitrogen into agricultural soils following planting cycles. So why glufosinate? Well, we chose glufosinate as our herbicide of choice because of its increased use in response to a rise in glyphosate or Roundup resistant weeds in agriculture. And so we can see a correlation in recent years of glyphosate decrease of use and non-glyphosate herbicide increase of use in figure 2. On the left, we can see a bit of a decrease in glyphosate herbicide use in the past 9 to 10 years. And on the right, we see a slight increase in the use of non-glyphosate herbicide in the past 9 to 10 years. And so glufosinate ammonium has taken the place of glufosinate as a common herbicide, and we see this as evidence in figure 3A, where our plant of choice, the soybean, is shown in the pink graph bars, and glufosinate ammonium usage has increased tremendously in soybean planting. And in figure 3B, we see a map of glufosinate ammonium usage across the country, and there are definitely some spots in Texas that use this herbicide a lot. And so the materials and methods that we use to test our objective of B. japonicum possibly being a bioremediation microbe in glufosinate-containing media with a growth curve experiment and a Kirby-Bauer assay. And so the growth curve experiment we conducted with Bradyrhizobium japonicum growth in 4 different concentrations of glufosinate ammonium solution. And all of this data was analyzed and visualized using Microsoft Excel, as well as our package's growth curver and ggplot2. And we see these results in figure 4 in a graphical representation of B. japonicum growth in the different concentrations of glufosinate. And so the sections where there are logarithmic growth of B. japonicum include our bacterium as well as 5 different concentrations of glufosinate ammonium. And the sections where there is stagnant growth or a flat line have no B. japonicum but do include the glufosinate ammonium in the solution. And so in this figure, we can see pretty much no change in any of the growth curves that we visualized. And in table 1, we have our some further data that we collected from our R packages. We have the carrying capacity, the initial population size that we began our experiments with, the growth rate, and the inflection point. And all of these numbers across the board have practically no change between all of the concentrations of glufosinate we used. And so this kind of shows us that B. japonicum growth is not affected by high concentrations of glufosinate. And so we used Kirby-Bauer to kind of visually confirm our results. And here we visualized absolutely no zone of inhibition with any of the glufosinate concentration punch paper that we added to our media and our bacterium. And so all of this data brings us to our conclusion that B. japonicum USDA 110 shows tolerance to high concentration of glufosinate ammonium. And with this, we would like to take our future work into finding the minimum inhibitory concentration by increasing the concentration of glufosinate ammonium we grow our B. japonicum in. Thank you so much for listening to me speak today. If you have any questions, you can leave them in the comments and I will respond accordingly. Thank you so much. I hope you have a wonderful day.